This video reports on research being conducted at Iowa State University on estimating and visualizing spatial cumulative distribution functions, known as CDFs, with particular applications to forest health monitoring. Members of the research group include Jim Majure, Dianne Cook, Noel Cressie, Mark Kaiser, Soumendra Lahiri, and Jürgen Symanzik. The aim is to provide a platform for interactive exploratory analysis of spatial CDF estimates through a fast mouse-controlled graphical environment. This is provided by linking a geographic information system, ArcView, and a dynamic graphics program, XGOBI. The data used in this video are from a sample of the forests of the northeastern United States collected by the Environmental Monitoring and Assessment Program, known as EMAP, of the U.S. Environmental Protection Agency. The sample includes the states of Maine, New Hampshire, Vermont, Massachusetts, Rhode Island, and Connecticut. The sampling grid is the hexagonal grid established by the EMAP program. Sample locations are shown in red and only include those sites that are forested. The variable being considered here is the crown defoliation index, which is an indicator of the tree crown response to stressors in the environment. The index is a weighted average of two variables, crown dieback and foliage transparency. Crown dieback refers to the mortality of relatively new branches that are in the upper, sunlight-exposed portions of the crown. The premise is that these branches have died from some stress other than the normal branch mortality that occurs from shading. The range is 0 to 100% in increments of 5%. Foliage transparency refers to the amount of sunlight that passes through the foliated portions of the tree crown. Transparency ignores any tree holes due to lack of branches. It is also measured on a range of 0 to 100% in increments of 5%. Because these variables are somewhat species dependent, we have stratified by species, and for the example used here, we studied only red maple trees. The points shown in red include only those sites at which red maple trees were found. The program is started by clicking the Go icon above the map on the ArcView window. This initiates the link to XGOBI. A variable is selected from a menu of attributes. In this case, the crown defoliation index is selected. Inclusion probabilities can also be specified for CDF estimation. The XGOBI window shows the CDF computed for the entire sample contained in the map view. Although the CDF in this case is univariate, the link can display two-dimensional CDFs, and the potential exists for displaying higher dimensional CDFs as well. Computation of the CDF is done as the data are passed from ArcView to XGOBI. The link is bidirectional. Users can interact with the map view in two ways. One way is to delineate spatial regions for which CDFs will be estimated. The second way is to brush spatial subregions for identification in CDF estimates. The user can also brush indicator values in the CDF plots for identification in spatial regions. This first example delineates two regions in the map. Sample points from the state of Maine are selected as one group, and the remaining states, New Hampshire, Vermont, Massachusetts, Rhode Island, and Connecticut, as a second group. The CDF for the Forest Health Indicator for Maine is colored orange, and the CDF for the remaining states is colored purple. Up to 10 regions can be specified for CDF estimation. The two CDFs differ noticeably in the middle. In this region, the CDF for Maine has higher values than the CDF for the remaining states. This indicates that Maine has a higher proportion of lower indicator values. That is, a higher proportion of healthy trees at sampling sites than the remaining states. Brushing in the subregions of Connecticut and Rhode Island indicates that these two states have high proportions of points with medium to low values of the index. Points corresponding to this subregion are indicated by larger glyph values in the CDF view. One of the main objectives of EMAP is to identify regions of the U.S. that have nominal or good, marginal or subnominal or bad health. These categories can be interpreted on a CDF as particular cutoff values. In the case of the indicator shown here, nominal corresponds to low values. In other words, small percentages of crown dieback and foliage transparency. And subnominal corresponds to high values. To identify locations which have subnominal health, we brush on high values of the indicator in the CDF view. The corresponding sample locations are highlighted in red in the map view. There appears to be a lower proportion of these values in Maine than in the remaining 5 states. One strength of the link to ArcView is the ability to quickly overlay geographic features on the map view. Here we have displayed county population densities. There does not appear to be a direct relationship between high values of the index, shown in red, and the population density, although there does appear to be fewer sampling sites in areas of high density. In this video, we have shown the application of a link between a GIS and a dynamic graphics program to the exploration of forest health monitoring data using CDFs. The link allows the data analyst to use the strengths of the GIS to overlay geographic features on the map and the power of the dynamic graphics program for manipulating high-dimensional data in a symbiotic manner.